Hi, I'm David, and this is the Biology Classroom. This is a paper discussion video, so get ready with this paper and let's go through the questions and answers together. Question 1 is about potatoes and Benedict's test. During storage, the starch content of potatoes decreases as they are broken down into glucose. A glucose assay can be used to determine the concentrations of glucose in potatoes. Let me make a summary of the test. First, potato juice is prepared for Benedict's test. Precipitates formed are removed from the solution. The filtrate contains copper ions that did not react with the glucose in Benedict's test. A calorimeter is used to measure the absorbance of the blue filtrate. The idea is that if there is a high concentration of glucose in the potato juice, most copper ions should be reacted with them to form the precipitate. Since there are not many copper ions left, the blue coloration would be lesser, giving a low absorbance reading. On the next page, we have a picture of a calorimeter and some brief information about how it is used. A1. Outline how calorimeters should be prepared before carrying out measurements on samples so that correct absorbance reading in the glucose assay are obtained. Before we can use a calorimeter, we need to calibrate it. This is to teach the calorimeter what kind of solution is considered zero. It will then compare all the other solutions with this first solution and generate values for them. In this experiment, water can be used to calibrate the calorimeter. We do so by using the calorimeter on distilled water and setting the absorbance to 0. Another thing that is usually done is that we can use a colored filter. This is to set the wavelength we want to use in the experiment. We will use a filter at the opposite end of the color spectrum with the solution so that the solution will absorb instead of reflecting the wavelength. Since the solution is blue, we will use a red or orange filter. In the next question, you are asked to show how solutions with different concentrations are prepared by proportional dilution. Do note that if you mistakenly use serial dilution in your answer, you can only get 1 mark maximum. Now, we have a 2% stock solution. We need to prepare a few other solutions, 20 cm³ each. First, list the concentrations we will use. You should have 5 concentrations that are evenly spaced. So, I choose 2, 1.5, 1, 0.5 and 0%. There are 5 solutions altogether, and they are 0.5% apart. Do not forget to include the units. To show how they are prepared, the best way is to use a table. I find it clearer and quicker. In this table, you must include the volume of the stock solution needed and the distilled water added to gain full credit. Make sure they are all 20 cm³ in total, as that is the requirement of the question. 3. Sketch the graph of the expected calibration curve that the student would obtain. The x-axis should be the independent variable, glucose concentration. The y-axis should be the dependent variable, absorbance. There is a negative correlation between these variables. Both a curve and a line are accepted for this case. B1. Identify the dependent variable in this investigation. It is the glucose concentration or the absorbance. B2 wants you to describe a method using the glucose assay to investigate the effect of storage time on the glucose concentration of potatoes. Details of how to carry out the glucose assay and how to prepare and use the calorimeter should not be included. There are a few things to take note before we start. The question is asking for storage of potatoes, so you should not mention storing potato juice. Besides, do not give vague statements. For example, when you want to talk about the storage temperature, you should give a specific value rather than just saying that they are stored at the same temperature. Lastly, remember to include the 6 components in the procedure writing question. Let's start with the steps. Potatoes are obtained from the same farm. This is to ensure the growing conditions are the same. Next, provide the independent variable. Always choose 5 specific values for this. Then, provide the storage condition of the potatoes. These are the control variables. For example, the temperature and dark conditions. Prepare 3 cm³ of potato juice from each group for glucose assay. Do not use a large volume which cannot be held by a test tube. That would be considered an illogical answer. Make a statement to describe how the dependent variable is measured. Since the question has mentioned that the details of glucose assay are not required, do not spend time writing them. To increase the reliability, make 3 replicates for each storage time. Calculate the mean and remove any anomalies. Do not use the word average. The correct mathematical term is mean. Make a safety comment with a hazard and a precaution. For example, Benedict's test involves boiling water bath, which may cause injury. Eye protection should be used during the experiment. There is a mark for AVP. For example, the same calorimeter must be used for all assays. In the next part of the question, we have some information about acrylamide, a substance formed when potatoes are cooked at high temperatures. It is toxic and increases cancer risks. In an investigation, scientists exposed raw potatoes to gamma radiation, which can decrease the acrylamide concentration of potato chips. They measure the concentration of glucose, protein, and volatile nitrogen compounds when potatoes are exposed to different doses of gamma radiation. C. The scientists suggested that gamma radiation reduces the acrylamide content of potato chips by affecting asparagine. Explain how the information provided and the data in Table 1.1 support this view. The question mentioned that volatile nitrogen compounds are present in potatoes due to the breakdown of some proteins and amino acids such as asparagine. So, if gamma radiation does cause the reduction of asparagine, there will be fewer volatile nitrogen compounds. The data shows us that as gamma radiation dose increases, volatile nitrogen compounds decrease significantly. Since there is no change in the percentage of glucose, the decrease in acrylamide is not due to glucose but asparagine. A decrease in volatile nitrogen compounds indicates a decrease in asparagine. So, less acrylamide is made since asparagine is needed to form them. In another study, scientists investigated whether gamma radiation and hot water treatment reduce the acrylamide concentration of potato chips. They used 4 doses of gamma radiation again, but this time there was treatment 2 where potatoes were cooked in hot water. 1. Calculate the percentage decrease in the mean acrylamide concentration of potato blocks given treatment 2 compared to potato blocks given treatment 1 for a gamma radiation dose of 0. To find the percentage decrease, find the difference between the two. Divide it by the initial value, and then multiply it by 100. The data was used to plot a table. Error bars were included. The scientists concluded that a combination of gamma radiation and hot water treatment is the most effective way to reduce the acrylamide concentration of potato chips. You have to explain how the information supports or does not support this conclusion. If your answer is only from one side, you can get 2 marks at most. So, you have to answer for both sides, as some data supports and others are not. Let's first look at the part that supports the conclusion. First, gamma radiation and hot water treatment decreases the acrylamide concentration of potato blocks more than gamma radiation only. We know this by comparing the whole treatment 1 and treatment 2. The error bars for treatment 1 and treatment 2 do not overlap at all doses of radiation. This shows that treatment with radiation and hot water is significantly better than treatment with radiation alone. Lastly, hot water treatment and 150 joule per kilogram gamma radiation dose give the lowest acrylamide concentration in the investigation. Now, let's discuss the parts which does not support the conclusion. First, hot water treatment alone decreases acrylamide concentration. We know this by comparing treatments 1 and 2 when there is 0 dose of gamma radiation. Secondly, error bars for treatment 2 overlap at all gamma radiation doses. This suggests that different doses of gamma radiation do not make a significant difference in treatment 2. We can also talk about the safety concerns. Hot water treatment alone might be safer than gamma radiation, so there is no reason to go for gamma radiation treatment. Only one variety of potato was investigated, so the data may not be sufficient to conclude that it is the most efficient way for all potatoes. Besides, there was no statistical test carried out for comparison, so the validity of this comparison is unknown. Question 2 talks about the GM rice plants. A concern about the plants is the gene flow between them and the weedy rice plants. Some scientists investigated the gene flow from herbicide-resistant GM rice plants to weedy rice plants. The hypothesis was that the gene flow decreases as the distance between them increases. Figure 2.1 shows how those plants were grown for the investigation and the direction of the wind. After pollination, the grains were collected from the weedy rice plants. They were collected from all the directions shown in Figure 2.1 and at different distances. The plants were grown and then determined if they had the gene for herbicide resistance. The development of herbicide resistance shows the gene flow between the GM plants and the weedy rice plants. A1 identified the two independent variables in this investigation. Both the distance of weedy rice plants from the GM rice plants and the direction from GM rice plants are independent variables. 2 wants you to outline a method to determine how many of the young weedy rice plants had the gene for the herbicide resistance without the extraction of nucleic acids. We can apply herbicide to the young plants and count the number of young plants that survive. Figure 2.3 is a graph showing us the percentage of gene flow recorded at each northwest collection point versus all the other directions. It also shows the percentages at different distances. We need to state two conclusions from the data. The first thing we can notice is that as distance from GM rice plants increases, the percentage of gene flow decreases. It is true for both lines. The percentage of gene flow is higher in the direction of normal wind or northwest direction than in other directions. We know this as the solid line is generally higher than the dashed line. Lastly, pollen cannot disperse further than 10 meters, as the graph shows that there is no gene flow at 10 meters or further for both the lines. In another investigation, scientists investigated both the gene flow from the weedy rice plants to the GM rice plants and vice versa. Table 2.1 shows the percentage of gene flow for the two situations. Question 1 wants you to calculate the mean. The answer is 0.036. 2. The scientists use a t-test to compare the percentage gene flow for the two cases. The null hypothesis is that there is no significant difference between the two, and the calculated t-value is 9.043. Table 2.2 shows the probability table for the t-test. We have to make a conclusion for the test. First, we must identify the critical t-value. To do this, you need to know what the degree of freedom in this case is. The degree of freedom is calculated by n1 plus n2 minus 2. n1 and n2 are the sample size of each group. It is 8 for the both of them. So, the degree of freedom is 16 minus 2 equals to 14. Remember that in a biostatistical test, the typical probability we use is 0.05, unless stated otherwise. From there, we can determine the critical t-value, which is 2.145 in our case. Since the calculated value is greater than the critical value, the null hypothesis is rejected. So, there is a significant difference between the percentage gene flow from GM rice plants to weedy rice plants and the percentage gene flow from the weedy rice plants to GM rice plants. 3. A student who read the paper concluded that the results show that there was no reasons to be concerned about gene flow from GM plants to wild plants. Suggest one reason why the results of this investigation should not be interpreted this way. We can answer this question by using the data directly or by discussing the limitations and weaknesses of the investigation. If you want to use the results directly, you can say that the data shows the gene flow from GM plants to wild plants does occur. Just because it may not happen that easily, we can't dismiss the effect, because even a low gene flow will transfer herbicide-resistant genes from the GM plants to the wild plants. Those wild plants to which the genes are transferred from GM plants may have a selective advantage. They have a higher chance of survival and can increase their number afterwards, causing disruptions to the ecosystem. There are also reasons to do with the limitations of the study. The investigation was only carried out once without any replica, so the data may not be sufficient. This investigation was only done on one crop species, so we can't use it to generalize for all GM plants. Besides, the fact that this investigation only tested 2 genes and in one geographical area makes it impossible for us to conclude for all GM plants. This investigation is only in experimental trials, so we won't know what it is like in real life. There is no assessment of the significance of consequences and no long-term data, so we do not have sufficient information to decide whether we can neglect the negative effect. That's all for today. If you think my videos are useful, don't forget to like, comment and subscribe. Thank you for watching and see you again soon.